Hello, good afternoon, everyone. Hope you had a good weekend.
Afternoon.
We are going to continue with the rest of lecture 2 today. I originally planned to start lecture 3 as well, but my morning class wanted to have some review for the quiz, um, which is on Thursday, of course, as you know. So I think that's what we're gonna do. We will finish up lecture 2, and then I will do a quick review for you guys as well. All right, so let's get started. This is the concept checking questions that we didn't finish last class. Let's take them up to begin. A peptide bond forms between which groups? So of course, this is talking about protein, right? And in proteins, the monomer is an amino acid. So let me just quickly draw the amino acid for you to remind you what the key components are. And as I mentioned before, you will not be asked to do any drawings on the test, on the quiz, but you should recognize an amino acid when I show you a diagram of it, okay? So the R is the side chain, and that's the only thing that is different between each of the amino acids. And this group right here, that is called the carboxyl group. Okay? And over here we have an amino group. I'm just gonna circle it on the other one here. Amino group. And so to connect the two amino acids together to form a bond, you will need to do dehydration synthesis, right? You should be familiar with this concept now, because dehydration synthesis is the process that we connect anything in macromolecules, right? So you will remove an OH, you remove an H, and you will form a connection here. Let me just do that for you. Remove the OH and remove the H, and then now I'm gonna have a peptide bond, right? Peptide bond. So what is the correct answer? Hopefully, you know it's C, right? Carboxyl group and amino group. All right? Just stop me if I am not making sense, if you want clarification. All right, let's take a look at the next one. Hemoglobin is an important protein that is composed of 4 polypeptide chains arranged in 3D shapes. What level of protein structure is illustrated by hemoglobin's 4 polypeptide chains? Okay, so there are 4 different levels, right? Primary, let me just refresh your memory, right? Primary is talking about the sequence of amino acid. Okay, so like the balls, right? Like the amino acid connects together. Now that is held together by covalent bond. Covalent bond. Someone asked me this morning, what's the difference between a covalent bond and a peptide bond? So in the context of macromolecules, right? They are polymers, and the polymers are connected together. Sorry, the polymers are formed by connecting monomers together, right? So when you take a glucose, connect to a glucose, the bond that holds them together, that's a covalent bond. When you take a glycerol and connect it to fatty acid, that's a covalent bond that holds it together. When you take an amino acid and you connect them together, that is also a covalent bond. All right. So covalent bond is basically what holds the monomers together. But we give it a special name when we are talking about proteins. Okay, so the covalent bond is a special kind because it's found in protein, so we call it a peptide bond. Okay, in protein. Okay, so peptide bond is a type of covalent bond. I hope that's clear. Secondary is they form these helix, right? And zigzags. Right, so that's the alpha helix and the beta sheets, and that's held together by hydrogen bond. Hydrogen bond from peptide backbone. Okay, so remember this is your backbone, and then you would have the side chains, right? R1, R2, R3, R4, so on and so forth, right? R46, right? R52, whatever, right? So the backbone is just I showed you last class, right? Like the NCC, NCC, so on and so forth, right? That is what will form the hydrogen bond and stabilizes the secondary structure. That's okay, Blessing. Okay, you can watch the recording after. Okay, hope everything is okay. All right, tertiary is when you have the three-dimensional shape, right? Three-dimensional shape, and that is held together by interaction of the side chains. Okay, interaction of side chains. So that's the overall 3D shape, right? Like you might loop back on itself, and now you have a 3D protein. Okay, and that is interactions of side chains. And sometimes you would have a quaternary structure where you would have multiple peptides coming together to form a complex, which would then perform a particular task, right? And so here, in the case of hemoglobin, the 4-polypeptide chain is referring to the quaternary structure. Okay, quaternary structure. As soon as you see multiple polypeptides that are involved in the protein, then you know it has a quaternary structure. Any questions? All right. Let's take a look at the next question. This is an example of an application question. I am going to launch the poll for you. Please select the correct answer.
Has the poll already blocked the question?
Are you able to move the poll window away? You should be able to do that, no?
Well,
Okay. Can you drag it away, maybe? No.
No.
Okay, sorry.
No,
Yeah, that's okay. Just choose something and then you can look at the question after. Yeah.
Okay.
And also, right, you guys have these questions, right? You don't have to read it off my screen, right? Like these are the PowerPoints that have been posted. Okay, so you can look at it on your printout, maybe. All right, it has been more than a minute and I only got 6 answers. Okay, I think I'm gonna end it. You don't want to spend like too much time on each question, right? Like try to allocate your time accordingly. All right. So this is not a particularly difficult question, but sometimes students, they read the first sentence, they're like, pancreatic cells make insulin. And then they feel overwhelmed because they're like, what is this thing? We never talked about that in class, like insulin, pancreatic cell, like what's going on, right? But it turns out this sentence is not necessary for you to complete the question. Okay? So like that you don't even have to understand what that means. What's important is the next sentence or next part of the sentence, which is a type of protein. Okay, so insulin is protein. That's what you need to know. These cells use something in order to synthesize protein by a process of something. So what is the monomer of making protein? You should know by now, it is amino acid. That's why the answer is E, right? That's all this is asking. It looks like it's that, and that's what an application question is, right? Like it throws you into a scenario that you haven't seen before, but really the underlying core question is something that you already know. I hope that makes sense. All right, here's another question. I will let you read it first, and then I will launch the poll. All right, let's see. Anyone else? Again, I only have 6 answers. Where are the rest of you, huh? All right. Now this one is like a sample quiz question, all right? Like a lot of people get this one wrong, actually. Um, and let's explain it, okay? Which of the following is false? So first one, the primary sequence of a protein is determined by the sequence of amino acids. So we know that one's correct. Okay, I wrote it down for you literally just 2 slides before, right? An example of secondary structure includes alpha and beta sheets. That's correct. Tertiary structures are stabilized by hydrogen bonds of the peptide backbone. Let's see what we wrote, right? Tertiary structure is stabilized by interactions of the side chain, right? Not the backbone. The backbone stuff is for secondary structure, right? Okay, so that is the incorrect one. The last one says increasing temperature will disrupt the secondary tertiary structure, but not the primary structure. That is true. Okay, so like the secondary, tertiary, and quaternary structures are quite weak. Okay, they are just hydrogen bonds mostly, and weak interactions. And so when you heat up the temperature, those things will unravel. So like think of the pipe cleaner I had, right? So this 3D protein, and when you heat it up, it will unravel. You know, secondary structure and primary structure will be gone, and then you'll be left with the bare primary structure. That you cannot destroy with just heating. Okay, like this is hard, right? Like I can't just pull it apart, right? So like you need a scissors to cut it, for example, right? So the peptide bond is strong, and the only way to break that apart is if you use hydrolysis, right? The chemical reaction to break it down. Temperature is not gonna affect it. Okay, so the correct answer is C. Some of these questions will show up on your quiz, right? So make sure you use them as practice. And that's it for proteins, okay? The last thing we're gonna talk about is nucleic acid, okay? Nucleic acid. This is a picture of chromosomes. How many chromosomes do we have? Does anybody know? How many chromosomes do we have in our cells? No, no guess? Give me a number. And any 32? Yeah, BB said 32. Okay, anyone else? 256. Okay, anyone else?
46.
46. Excellent. That's the right answer. Okay, you have 23 from each of your parents, and a total of 46 chromosomes. And the chromosomes are colored here using a technique called SKY, SKY. And this is a useful technique because it allows you to visualize the chromosome very easily. For example, you can see if part of chromosome 8 has been broken off and maybe attached to chromosome 4. I mean, it didn't happen here, but if it did, then you would see purple with a bit of blue, right, on number 4. Then you're like, that is a chromosomal abnormalities. And in fact, some forms of leukemia has these kind of abnormalities where one chunk of the chromosome breaks off and then attach onto another one. Okay, it's called a Philadelphia chromosome. You can search it up if you're interested in it. So, that's just what DNA is. In terms of composition, we have the triplet that we always have, the CHO, and also nitrogen as well as phosphorus. Okay. What other molecules have we learned in this lecture that also contains phosphorus? Does anybody remember? What other molecule contains phosphorus in this lecture? Quiz is on Thursday. You should probably know this. Good try. Not protein though, not protein. Looking for a specific molecule that we've learned that also has phosphate. Like what if I ask you on the quiz, right? Select all the molecules below that contains phosphate, then you have to know, right? Sebastian says ATP. Yeah, you're correct, but we haven't learned ATP yet though. Okay, so something that we've learned so far that it has a phosphate in it. It's even in the name. The name of this molecule suggests to you that there is a phosphate in it. It's a kind of lipid. Help me out here, please. What kind of lipids am I talking about? Phospholipids. There we go. Thank you, thank you. So that's the other molecule that has phosphate. All right. So nucleic acid has CHON as well as P. And the monomer of a nucleic acid is what we call a nucleotide. Okay, nucleotide. There are two main types of nucleic acid. There is DNA, which most people have heard of. And then before the pandemic, people usually don't know about RNA. It's like, not very common thing to mention. Okay, but then in the pandemic, right, some of the vaccines are made up of RNA, right? RNA vaccine, right? And people become more aware of it. Both of them are nucleic acid. Okay, DNA is deoxyribonucleic acid, and then RNA is ribonucleic acid. Deoxy means it has one less oxygen compared to RNA. Okay, that's why it's deoxy. I'll show you what that looks like in a bit. Let's take a closer look at nucleotide. The nucleotide has three components to it. There is a phosphate group. Okay, there's the P. It's actually PO4. Right? You will learn that in chemistry if you haven't already. It's connected to a 5-carbon sugar. By now, you should know we can represent sugar using hexagons and pentagons. So there is your sugar. Okay, and this sugar can be a deoxyribose, as in the case of DNA, or it could be a ribose, which is RNA. Now, the suffix OSE, O's, means it is a sugar. Think of all the sugar that we have learned so far, right? Glucose ends with OSE, fructose, maltose, galactose, lactose, right? Cellulose, right? All these things are sugar and they all end with OSE. So deoxyribose and ribose are just another kind of sugar. It's going to connect to a nitrogenous base. Okay, nitrogenous base like this. So this whole thing is a nucleotide. Let's draw another nucleotide here. Nucleotide, right? So this nucleotide and this nucleotide would form a bond, right, to connect it together. After all, this is a polymer. So you need to have nucleotide connected to a nucleotide connected to a nucleotide and form a long chain, right? So if you have been listening to me since the beginning, what kind of bond is this called? What kind of bond holds the nucleotides together? Good try, Justine. But peptide bond is specific for protein. Okay, I'm looking for like a more general. Yes, covalent. Very good. Covalent bond. All right. Is everybody okay? I feel a little bit uneasy. Um, should I be worried about Thursdays? Hopefully not, okay? But please, if you don't understand, ask. Okay? If you don't ask, I don't know what you don't know, right? All right. So then we can have another one here. Look, I can connect to another sugar here and then phosphate, and then you would have the nitrogen coming out, okay? Why not ionic? Okay, so all the bonds that we've learned in the macromolecules, they are all covalent bonds. Okay, they're all covalent bonds. Ionic is metal with non-metal, right? There is no metal here. Everything, all of these are just non-metals, right? Okay, so that's the short answer. All right, so here, this is called the sugar phosphate backbone. It's just, sugar, phosphate, sugar, phosphate, sugar, phosphate. All right, it forms the sugar phosphate backbone. Okay, and of course, to form these linkage, to form these bonds, you would have to do dehydration synthesis. Right? You're going to remove a water molecule. Right? It's the same concept, right? Over and over again. All right, that's that. Let's take a closer look at the nucleotides. This is a typical nucleotide on the left for DNA because it's deoxyribose, right? So this is DNA nucleotide. And this is RNA nucleotide. We have the phosphate. We got our 5-carbon sugar. And then we have the nitrogenous base. So take a look at the deoxyribose here and the ribose. They look pretty much the same, okay? Except for this part right here. It's like one of those spot the difference game, right? So you can see this is missing an oxygen, right? One less oxygen. That's why we call it deoxyribose. Okay. Now the base, the nitrogen base, is called nitrogen base because it has nitrogen on it. All right. So for DNA, it could be one of 4 things. It could be A, G, C, or T. All our genetic materials is made up of these 4 letters. Okay, we have literally like billions of these letters in every single one of our cells. Right? Our blueprint of life, right? They call it, is just 4 letters: A, G, C, or T. I'll show you what they stand for in the next one. For RNA, it's very similar. It's also A, G, and C, but it does not have the T. Okay, it has the uracil instead, the U. Okay? So that's the difference. A lot of information I'm going to tell you on this slide, so please pay extra attention. So these are the nitrogenous base, right? And it tells you what they stand for. So A is for adenine. That's how you pronounce it. G is for guanine. C is for cytosine, thymine, and uracil. Remember, thymine is only found in DNA, and uracil is only found in RNA. We can classify the nitrogenous base as either purine or pyrimidine based on the number of rings its structure contains. So purines include adenine and guanine, and they have 2 rings. Okay, I could not memorize this when I was learning it, for the first time, right? So eventually, this is how I remember it. Okay, if you remember, pure as gold, pure as gold. So purines are adenine and guanine. Everything else is pyrimidine. Also, purine has 2 syllables, right? So purine, 2 syllables. 2 syllables, 2 rings. That's how I remember it. Okay, you can come up with your own way of remembering it, but that's how I do it. So purines have 2 rings, and pyrimidine has 1 ring in the structure. All right. So keep this in mind because it will help us understand the structure of DNA in the next couple of slides, I think. Just a side note, there is a condition called gout. You might have heard of it. Right? It's common in older people, older like, about 40, I guess. And it's built up of uric acid in joints, particularly like the big toe, and it's really, really painful. So people with gout, they actually have to watch the diet and not consume too much purines. There are certain food that is high in purines, like seafood, red meat, and alcohol. Okay, so consuming those food will make the condition worse. It will hurt a lot more. So gout, if you didn't know, now you know. Okay, back to our regular schedule program. Here is the double helix, and I want you to take a look at the diagram. Do you notice any trends in terms of what letters always goes with what letters?
Yes.
What's the trend, huh? Yeah, right. T goes with A and C goes with G, right? A pairs with T and G pairs with C, right? So let's think about this without looking at the previous slide. G, is G a purine or pyrimidine? Is G a purine or pyrimidine, right? Hopefully you say purine, okay? Because then remember, pure as gold, right? So G is purine, and purine has 2 rings, okay? Cytosine is a pyrimidine, so it has only 1 ring. Let's take a look at the other one here. And it could be backwards, right? Like a G could be on this side and then 2 rings, 1 rings. Thymine? Thymine has 2 rings or 1 ring. 2 rings or 1 ring. That's right. It has 1 ring because it is a pyrimidine, and then A is a purine, so it has 2 rings, right? So this is important observation because by following the base pairing rule, the width of your DNA is going to be the same along the entire length. Okay, think of the DNA as a twisted ladder, right? Okay, so, oh boy, this is a ladder, right? So the rocks that you climb on, those would be like the ACTGs, okay? And the width is always 3 rings wide. If we don't follow the base pairing rule, then the DNA would like be bulging at some places and they will be like very narrow in some places. And you can imagine like this would be like not a very stable structure, right? It might break apart, right? So that's why the base pairing rule is important. Now the blue and the yellow alternating pattern, that is the sugar phosphate backbone. Sugar phosphate backbone. Okay, that's basically the sides of the leather. Okay, and then the rugs that I told you about, that would be the nitrogen base. Okay, so you take the leather and then you twist it, and you will get your double helix right here. All right. Hope I'm being clear. So that's the base pairing rule I told you about. And the fact that the DNA strands follow the base pairing rule means if you know one side of the DNA, you will be able to come up with the other side, right? Okay, if I tell you one strand, you can use the base pairing rule to figure out the other side. So this property makes the DNA complementary. Okay, knowing one strand is enough to predict the other strand. Maybe predict is not the right way. Like you don't have to predict, like you know what it's gonna be. If I tell you this side is AAC, right, then the other side must be TTG, right? Okay. If you pay attention to this picture, you will see that again, the backbone is sugar and phosphate, sugar, phosphate, sugar, phosphate, and that's true on both sides. The base pairing rule applies to the nitrogenous base. Holding the sugar phosphate backbone together is covalent bond. Nothing is new so far, right? There are two additional pieces of information that I want you to be aware of. First of all, if you take a look at this side of the DNA versus the other side, you notice they are upside down relative to each other, right? Okay. So it's like this. Think of like people lying down on the floor. Okay, and then you would have another line of people lying next to them. Okay, so one is like, head to feet this way, and then the other is feet to head. They are opposite but parallel to each other. So this property of DNA is called anti-parallel. Right? DNA strands are complementary and anti-parallel. One more thing to pay attention to. There's a lot of information. I'm sorry. You see these dotted lines, right? That's holding the A and the T together. These are hydrogen bond. Hydrogen bond. Right? By now, you have heard me say hydrogen bond a lot, right? So in case you're kind of not sure what that is, all you have to know is think of them as like tapes, okay? That help tape things together. Okay? So we have hydrogen bond in the peptide backbone that tape the helix and the sheets together. We have hydrogen bonds that tape the side chains together. We have hydrogen bond between water molecules that allows them to cling together. And now we have hydrogen bonds that allows the bases to link together. Okay, I should mention hydrogen bonds are quite weak. Okay, you can easily break the hydrogen bond by applying heat. That's why, if you remember that question with the protein, if you heat things up, you will disrupt the secondary, tertiary, and quaternary structure, right? Okay, so similarly, if you heat up the DNA, you can pull the two strands apart. So between the A and the T, there are two hydrogen bonds. Okay, two hydrogen bonds between A and T. But then there are three hydrogen bonds between C and G. Which means if you have a DNA molecule, right, a helix that has a lot of C's and G's in it, it would be harder to pull apart, right, because they would have more hydrogen bond. The opposite is true. If you have a strand that has more A's and T's in it, then it's easier to pull apart because there's only two hydrogen bonds holding them together. Does that make sense? And again, like you might wonder, like, who cares? Like 3 bonds, 2 bonds, what does that have to do with anything, right? So PCR. Have you guys heard of PCR? You must have heard of PCR, right? What do we use PCR for? Yeah, PCR. Yeah. What do we use it for? Like during the pandemic, what do we use PCR for, guys? Really? None of you have had a PCR test done? Okay, Sebastian says COVID testing.
Uh,
That's right. Okay.
yeah.
So PCR actually has been around for a long, long time. Okay. 50 years, I would say. But most people haven't heard about it until the COVID, right? So PCR is a way to clone DNA. We'll learn more about it. Yeah, exactly, right? They use it in forensics, right? If they find some blood samples at the crime scene, how do you know it belongs to which suspect, right? So they can use PCR, right? So we'll learn about that in lecture 9. But in one of the steps in PCR, you have to heat up the DNA, right? And to separate the two strands. That's part of how it works. So how do you know what temperature to heat it up to? Well, you have to know how many C's and G's are in there. Okay, if it has a lot of C's and G's, you got to heat it up more. If there's not a lot of C's and G's, then you don't have to heat it up as much, right? Okay, so that's like one example of why you might care. But sometimes we just have to learn things, right? Like not everything has to have a reason why you have to learn it, right? Like you just have to learn it because you want to get good grades and move on to your next program. But I try to bring in like real world examples that help you see a connection. I see relevance. But yeah, I don't know. Hopefully that piques your interest a little bit. Moving on, we have sequence here, okay? A, G, G, C, T, 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 so on and so forth. That's one side of our DNA. And using base pairing rule, okay, we are going to write out the other side. Now, you don't have to unmute because it's going to be very chaotic, but, say it with me, okay? So I can hear you through the computer screen, right? So with the A on one side, what is on the other side? Well, A pairs with T, right? And the next one is C, C, G, and then A, A, T, T, A, T, G, C, C. Right? Easy. If you know one side, you can predict the other side. This property of DNA is called complementary. Right? You know one side, you can figure out the other side. The other property of DNA is that they are anti-parallel. They go in opposite directions. All right. So this one, right? This one has a lot of A's and T's in it. So if I want to separate it, probably doesn't take a lot of heat. Okay. Just a little bit of energy will be enough to pull them apart. All right, let's do some math. All right, everybody gets a little bit tense when they hear what we have to do math in biology. Actually, we do, especially in the genetics unit later on. There is quite a bit of calculations. So let's start with some easy one here. Suppose a cell has 40% adenine. Okay, find the percentage of T, C, and G. Okay, I should say in its DNA. Okay, in its DNA. I mean, you kind of know I'm talking about DNA, right? Because this is referring to thymine, right? So if you have 40% adenine, right away, what else is 40%? What else is 40%? Yeah, T, right? Exactly. Because A pairs with T. So whatever amount of A you have to have the same amount of T, right? Okay, because the two goes together. All right. It's like if I ask you, okay, I found 40 40 left shoe, okay, in the gymnasium. How many right shoe are you gonna find? 40, right? Because they come in pairs, right? Okay. Whatever you have for A, you would have for T. I don't know. That analogy sounded better in my head. When I say it out loud, it seems a little bit weird. Hopefully it helped. Now, if I add these two together, right, 40 and 40, that makes 80, right? Okay. Now, I don't know if you know, but everything together is 100%. So if you take 100% and you subtract 80, that means I have 20% left, yeah? These 20% accounts for the remaining C's and G's. But C's and G's are together, right? So I have to take the 20 divided by 2, then I get 10 for C and 10 for G. Any questions for this?
Please, can you explain this? I don't understand.
Okay, sure. Which part do you have a specific
Um,
question?
the whole thing, like how the
The
whole
whole thing?
question.
All
Yes.
right, all right. Let's try different numbers, huh? Let's try different numbers. Uh, okay, let's say we have 35% C. Okay, 35% C. Okay, we're gonna find the rest, people. So, if I have 35% C, Tahib, maybe you can help me out here. What else is 35%? What else is 35%, huh?
Um,
What does
T.
C pair with?
Same pair with T.
C pairs with G, okay? C pairs with
G.
G, G, G.
Oh,
A pairs with T, okay? So that's the first thing you have to remember. What pairs with what, right? Because C pairs with G, so if you have 35% C, you would have 35% G as well. Is that okay?
like how come P pairs with C pairs with G? How?
How come C
Uh,
pairs with G?
yeah.
Oh, I mean there's no particular reason. Like that's just the way it is, right? Like if you look at over here, right? Like where is it? There's a slide that says it, right? Look, A always goes with T and G
Oh,
always goes with C so that you can
oh,
maintain the
okay.
width of the DNA, right? Remember? Okay. So C's and G's like this. So that adds up to 70%, adds up to 70%, which means you have 30% remaining. Divide that into 2, we have 15% A and 15% T. All right. So if anyone want to listen to this again, you would want to see the recording and it would be at the 40-minute mark. Okay. All right. Go to the YouTube recording and then skip to 40 minutes. This is where I explain these things. Okay. Just watch a couple times. You should be okay. All right. Moving on. Last slide, right? Besides DNA, the other nucleic acid is RNA, which is single strand. Okay, so it doesn't have two sides, it's one side only. The backbone has the ribose sugar as opposed to the deoxyribose. And it's made up of phosphate, just like DNA. In terms of the nitrogen base, it's ACG and uracil instead of thymine. Okay, uracil. There are different kinds of RNA. If you know about the vaccines, the COVID vaccines, that's the messenger RNA, mRNA vaccine. We also have rRNA, tRNA, to name a few other examples. Okay, so don't worry about what they do. We will learn it in lecture 7, I think. All right, let's do some poll questions. Again, I'll let you read it first, and then I will launch the poll. Okay, just a couple more seconds. Again, try to participate if you can. All right, even if you're not sure what the answer is, just choose something. All right, so that I know you're there. All right, I got 58%, 64% participation. Okay, that's not bad, I guess. All right, so let's take a look here. Which of the following is true regarding purines? Okay, so first one. First one. Adenines bonding to thymine is stronger than guanines to cytosine. That's not true because adenine to thymine is weaker. They only have two bonds, right? Only have two bonds, hydrogen bonds, as opposed to three bonds. So that is incorrect. Next one. Cytosine is a purine. Okay, remember, pure as gold. That's the phrase to remember, right? So purines are adenine and guanine, which means this is not true. Cytosine is a pyrimidine. Purines bind readily to deoxyribose, but not ribose. That's also incorrect. Okay, deoxyribose is DNA, ribose is RNA, right? So let's take a look at an example of purines, A and G. Both A and G are present in DNA and RNA, so they would bind readily to both kinds, not exclusively to one versus the other. Both adenine and thymine are purine. Nope, adenine and guanine are. So that's not correct. Purines have double ring structure, two rings, right? That's correct. All right. So some of the questions on the quiz will be like this. Okay, there is like, you have to read it carefully and to find the answers. Other ones would be relatively straightforward, like which of the following is an example of a purine, right? Something like that. It's factual, it's easy to find. And then these ones require thinking and analysis. Let's do another one here. This is an example of a multi-select question. It has more than one correct answers. I will, again, let you read it first. All right. I will end the poll. So let's take a look. Which of the following is not a feature of DNA? First one, nucleotide within a single strand are connected by covalent bond. That is true. Right, let me show you. Right, this is one strand of a DNA. This is the second strand, right? What's holding them together? Covalent bond, right? Covalent bond. So that is not the answer we're looking for, because we are looking for things that are not true for DNA, right? Adenine forms a base pair with uracil. That's not true. There are no uracil in DNA. It should be thymine. Complementary strands are held together by hydrogen bonds between phosphate group. That is incorrect. It is held together by the nitrogen base, right? Nitrogen base. Complementary strands are anti-parallel running in opposite direction. That's true. That is a DNA thing, right? Remember, I drew the people lying down opposite to each other? Right? So the correct answers are B and C. Right? So like I said, there will be 5 multi-select questions on the quiz. These are 2 marks each, and you have to select all the correct answer in order to get full marks. Otherwise, you will have part marks. All right, here's another example of an application question. Please try it out. All right, last call. Anything else? Anyone else wants to participate, huh? Okay, thank you. So let's take a look at this. A chemical reaction occurs slowly at 37 degrees Celsius. When molecule X is added, in other words, when something is added, the reaction speeds up significantly. Okay. What do we add to speed up a chemical reaction? What is the molecule? What kind of molecule have we learned? Right? Justine said capital enzyme. That's right. That is enzyme. Right? Enzymes are protein, right? That speeds up chemical reaction. They catalyze chemical reactions by lowering the energy requirement, right? Remember I said it's like scholarship that lowers the tuition fee. Okay. So once we have figured out that it is enzyme that they're talking about, then the question becomes much more manageable. Which of the following is a monomer of protein? Okay. So you should recognize what this is, right? This is amino acid, right? What about this one? What is this molecule? Anybody recognize this? You know, there is a phosphate, there is a sugar, there is a nitrogen base here. We literally just learned this. What do we call this? Well, I mean, like, yeah, we don't know if it's a DNA per se, but you know, it's nucleotide for sure. Okay. And then this is a hexagon, so some kind of sugar. And this is fatty acid, right? So what makes up protein? Well, amino acid. That's how you would approach this question. So that's it. That's it for lecture 2. I know it's a lot of stuff. We're like what 106 slides, right? So like a lot of things to study for. Okay, I just want to reiterate you can use your notes on Thursday, right? Okay, but you won't be able to access them through your computer. I don't know if I told you that already, right? Since you'll be using a lockdown browser. So if you need to have your notes, you got to print them out or you got to like, write it out and put it right next to you. Okay, you won't have access to the online notes during the test. Yeah, what do you mean? Like, as in, like, can you use the study guide? Is that what you're asking me, Justine? Yeah, you can use whatever you want. Okay, I don't care, okay? Well, you cannot have another person do the test for you. That's not allowed, okay? But you can use whatever notes. Yeah, you can use the study guide. You can print out the one that has the answers in it. Doesn't matter, right? Okay. But the thing is, like, you have to have physical copies of them, right? If you're gonna use it, because once again, when you start the quiz, your computer will be locked down. You can only do the quiz, okay? And it will remain locked until you finish the quiz. All right. Any questions about the quiz before I do a quick review for you? Any concerns? Things you want to ask me? Ask me anything. I'll tell you.
Can we talk about monomers?
Yeah, I will. I will give you a summary in a second, okay?
Okay.
Yeah. But concerning the quiz, how you take it, you take it online, right? Yeah, you see the link I gave you, right? And let me see right here. Uh, you guys are like what? 202? Yeah, there it is. So I posted an announcement, right? That's the quiz link, right? Okay. And many of you still have not installed the lockdown browser. Okay, you gotta do that. You have to do that. Okay, so go back to like what is it? Like um I forgot, like last week here, maybe, no, even earlier. Week one, right? Week one, Tuesday, right? You click on the scientific method self quiz to install the lockdown browser. I cannot stress the importance of doing that before Thursday, okay? Because things can go wrong and you want to be able to fix it before you do your actual quiz, okay? We have a class on Thursday at 12:30. Okay, so you can do your quiz either before class or after class. The quiz will be available to you from 6 a.m. to 11:59 p.m. All right. Now, I like you guys. You guys seems to be good students. So I'm gonna tell you the short answer question on the quiz. Remember, the quiz has two short answer questions, 17 multiple choice and 5 multi-select, right? Okay. So get ready. One of the perks of coming to class, you get to see what's on the test. So the first question, these are questions you have to type the answer for, right? Okay. So the first question, I'm gonna give you a picture. Right? I'm not gonna tell you a picture, but I'm gonna give you a picture, and I'm gonna ask you to describe using qualitative observations. All right? That's gonna be one of the questions. You guys remember qualitative observations, right? If you don't, please watch the recording again. So that's the first question. All right? The second short answer question. I'm gonna give you a scenario, okay, where you have to perform an experiment to test something, okay? And then I will ask you what is the dependent variable, what is the independent variable, and then what are some standardized variable, okay? That's gonna be the second question. All right. So the second question I think is worth four marks. First one I think is like two or 3 marks. I don't remember. Okay, but those are the long answer question, short answer questions. Okay, the ones that you have to physically type in. All right. Don't just copy what I have in the notes, right? Like write it in your own words. All right. And you should be okay. Yeah, you're welcome. All right, no problem. Well, if there are no other questions, let's do a review. Okay. Now, you guys might like I said, right? Like you shouldn't be relying on your whole pack of notes, right? Because that's too much, right? Like you gotta flip through it, you gotta find the slides, so on and so forth. What I recommend is creating like a cheat sheet, right? Like a summary or something that would help you. And in the process of creating that help sheet, it actually helps you study, right? Like I was telling my morning class, like you shouldn't be reading biology notes as a way of studying. Okay, reading it is not going to help you. You have to write it out. I recommend taking a piece of paper and then like think of the topic like carbohydrates or something. Write out as much as you can remember for carbohydrates, and then go back to the notes to find the things that you've missed, and then add it to your notes with a different color. And then, the next day you do it again. Hopefully you will remember more, right? And if you repeat this process a few times, it will help you retain the information. But if you don't know how to start, have a writing your help sheet, you can use this one. Okay, I'm gonna write it out for you right now. I strongly recommend you copy along with me. Okay, I'll post it online after in case you missed something, but please write it along. So let's start with like carbohydrate or something. Okay, I'm not gonna have time to review lecture 1. Okay, but let's focus on lecture 2 first. Right, so carbohydrates. It's made up of C, H, and O, right? Carbon, hydrogen, oxygen. And we have the monosaccharides, right? Monosaccharides, which include things like glucose, galactose, and fructose. And that's what I mean. You should sit down and try to write this out on your own from memory to see how much you remember, right? And then you can combine the monosaccharides together to get disaccharide. Okay, so like, glucose plus glucose gives you maltose. Glucose plus galactose gives you lactose. Glucose plus fructose gives you sucrose, right? So these are disaccharides. These are monosaccharides. And then we can put many, many glucose molecules together using dehydration synthesis to make polysaccharide. Okay, so help me out here. Help me out. What are some examples of polysaccharides? We have 4. Okay, just type them in the chat box, yell them out. What is it? Show me you've learned something from me. Cellulose, that's good. I'm gonna put it right here. Anything else? Starch, good. Glycogen, yeah. And also chitin, right? Chitin. Right? So these are for structure, right? These are for energy storage, right? So cellulose, they are like the fibers in plants, right? Cell wall of plant cells, right? They provide structural support in plants, essentially. And chitin, they form exoskeleton of insects and crustacean, right? I can never spell this right. Crustacean with the A, I think. Okay, so these are like, you know, lobster and crabs and stuff like that. Sorry, crustacean. The hard part, right? That's chitin. Starch is the storage form of energy in in plants. You will find them in roots or stems. Think potato, think yam, right? Cassava. Those are roots and stems of plants. What about glycogen? Where do we find glycogen in the body?
In
Anybody?
the muscles,
Yep, in the muscle and where else? Remember, I had to stand up to show you where the other thing is?
the liver,
Yep, the liver, right? Which is just below your diaphragm, right? On the right side, right? So, we have the polysaccharides, we have the function. That's pretty good, right? Another thing that just came to my mind, right? And I'm not doing this in any particular structure, and you should, I mean, you should when you do your study notes, I guess. But it's like whatever comes to your mind, right? Like so, for example, glucose. We need this for cellular respiration, right? Which is like glucose plus oxygen produces carbon dioxide and water, right? And energy. And energy. Okay, so that's a nice summary of carbohydrates, I feel like, right? That's everything that we learned for carbohydrates. Anything that you want to add to it?
sorry, I missed the part. You were saying glucose plus oxygen for cellular respiration,
Mhm. What
and
what
then
about?
they produce carbon dioxide and water.
Yeah, carbon dioxide, water, and energy, right? Hmm. We had a slide on it. Let me see if I can find it. Somewhere in here. There, right? Glucose plus oxygen produce carbon dioxide and water, right? It's the primary way of producing energy in the body. Okay, so that's that. Let's move on to our next macromolecule, which is lipids. Okay, lipids. So lipid has CHO and P for phospholipid. Okay, P for phospholipids. So lipids are different because it doesn't have monomers, right? Instead, it has components. So some of the key components include glycerol, not to be confused with glycogen. Okay, this is glycerol. Fatty acid, which could be saturated or unsaturated. If you forgot what that means, I'll explain in a second. You have a phosphate group, phosphate head, and carbon rings. Carbon rings, right? And then sometimes you can have functional group as well, depending what you're building, right? So, triglyceride. Triglyceride is fat, and to build fat, we need to have glycerol plus 3 fatty acids. What do we need fat for? Long-term energy storage. Insulation for organs, right? So let's talk about the difference between saturated and unsaturated fat. Okay, so first of all, this is not so healthy, and this is healthier. Okay, I wouldn't go like drink a cup of olive oil necessarily, but then it's the healthier choice. So what have we got here? For saturated fat, it is 1 glycerol plus 3 saturated fatty acids, right? And what does it mean to have saturated fatty acids? No double bond, no kinks. Kinks are like, bands, right? No sharp turns. And they are bound to maximum number of hydrogen atoms. No more space. That's why they're called saturated. You can't fit more hydrogen, even if you wanted to. Are these solid or liquid at room temperature, huh?
Solid.
They are mostly solid because they pack closely. Solid at room temperature. Most of them are animal derived. Okay. E.g. butter, lard. On the other hand, for unsaturated fat, it is 1 glycerol plus at least 1 unsaturated fatty acid. Okay, you can have 2 unsaturated fatty acids, you can have 3. The more you have, the healthier it is, right? It is called polyunsaturated fat. Okay, so here it has it might have 1 or more double bond. Okay, some kinks. All right, so in class I was like telling you they look like this, right? With the kink. So they don't pack together easily. Um, they still have room for more hydrogen. And then these are liquid at room temperature and plant derived, mostly. Okay, there are exceptions, right? I told you in class. So like canola oil, right? Olive oil, whatever oil you like to put, peanut oil. Done. Okay. And we talk about how we can convert from unsaturated to saturated, right? Using hydrogenation. Right? You force them to take in more hydrogen and they become spreadable. That's like margarine shortening. All right. Now, other than that, we also have another kind, right? Phospholipid. Which is glycerol plus phosphate head, phosphate group, same thing, plus 2 fatty acids. They could be saturated, they could be unsaturated, could be a mix, doesn't really matter. What does matter is you have this molecule, right? Okay, that's hydrophilic head and hydrophobic tails. And it's the major component of what? What do we use phospholipids for? It's a major component of. Okay, that's fine. Major component of cell membrane. Right, so these are kind of things to remember. Like, could you look that up during the quiz? Yeah, sure, but then like you waste time, right? There are only 45 minutes for the quiz. Okay, it's not a lot of time. There's wax in the notes, but there is no question on wax in the quiz. You don't have to worry about it. So we just skip that. And the last one is steroid, right? Okay, steroids are 4 fused carbon rings plus functional groups, which gives it unique properties, right? Okay, so example is cholesterol, which we can use for various function. Okay, it's also another component of cell membrane. Okay, it is needed to make biosalt, right? Vitamin D and hormones such as testosterone, estrogen, right? That's the lipid stuff. Any question for lipids? I hope this is not unfamiliar to you, right? Like if it feels like this is the first time you're hearing it, then we have a problem, right? Like we've learned all these already. Okay. So make sure you review them. All right. Then we have proteins. All right. So protein is CHON, and nobody could answer me this morning. Maybe you'll do better. Trace of what? In addition to CHO and N, there is one more thing. Yes, sulfur. Thank you. Trace of S. Okay. So we know the monomer is 20 different amino acid. 9 are essential, right? That means you cannot eat them. Sorry, you cannot make them. You have to eat them. Okay. You already know the structure. I mean, I can draw it again. Hopefully you recognize it by now. You must be sick of seeing it since I wrote it so many times. Okay, so again, we have the carboxyl group. We have the amino group. Okay, we got the side chains here. Right? And you can connect two amino acids together through dehydration synthesis. Okay? And it forms a covalent bond, but it's special, so we give it a special name. It's peptide bond. Now, I don't want to rewrite this thing. It's in the study guide answer, but you should probably familiarize yourself with it, right? Again, the different levels of structure. And I mentioned earlier to you that The last three levels, the secondary, tertiary, and quaternary, these can be disrupted by temperature, salt concentration, pH, right? This one will not be affected by any of these, okay? Need hydrolysis to break peptide bond. Okay? So heating a protein up will only disrupt the last three levels, right? And when you do that, you are denaturing the protein, right? Denaturing the protein. Okay? And denature the protein. Oops. Functions. Okay, what do we need proteins for? Structure. Okay. Muscle, hair, horns, nails. Okay. Just put down some examples. We also need proteins for enzyme and hormones, right? Some hormones are steroids, like the estrogen, testosterone. Some hormones are protein. The only example I gave you is insulin. Okay, you might know others, fantastic, but that's the only one we've done. Okay? So let's have a word about enzyme. Enzyme, they catalyze reaction by lowering energy requirement, right? It's called activation energy if you look at the notes. So like, that there's an enzyme here, enzyme, and then it has these little pockets. Those are called active sites. Okay, and then the pocket would allow you to fit some molecules in it, right? This is the substrate. Okay, so the substrate would fit into the enzyme. And again, there is a key and lock relationship. So the enzyme will only work for a particular substrate. Okay, if I have another substrate that looks like this, okay, like this is pointy, it will not fit into the active site. So this would need like another enzyme, right? So one enzyme typically only works on one substrate. Okay, and then it will carry out the reaction, break the bond, split out the product. All right. I think that's most of the protein stuff. And then today we learn nucleic acid, right? Nucleic acid, CHO again, N and P. Okay. We literally just learned it, so I don't want to write too much. I'll do a quick comparison between DNA and RNA though. So for example, we can talk about the nucleotide. Okay, so nucleotide, sorry. I meant to say monomer. Okay, so monomer here is a nucleotide. Okay. Same thing here, nucleotide. The kind of sugar that you will find in the nucleotide would be deoxyribose for DNA and ribose for RNA. The base, ACGT, ACGU. Right? Uracil instead of thymine. Structure. This is double helix. This is single strand. That's all. That's all the difference there are. Okay, so you can add to it after class. Right? Write down the purines, the pyrimidines, the number of hydrogen bonds, pairing rules, and so on and so forth. Okay. Any other questions? All right, so let's do practice, right? With like a scenario. Let's say you want to test if different running shoes allow you to run faster. Okay. Let's say that's the scenario, right? So I told you you're gonna have a scenario question on the quiz, right? So what's the hypothesis? Okay. Why don't you type in hypothesis for me? Okay, just type in the chat box. Don't press enter yet. Type it. Okay, and then we'll press enter together. Okay. Go ahead. Participate. All right. All right. If you have an hypothesis, please press enter. I want to see it in the chat box. Only one? Oh my goodness, you guys hurt my feelings. Okay, we have like two. Okay, three. Okay. Man, online teaching is tough, but that's okay. That's okay. I mean, you know, if you type one, fantastic. All right. So this is the hypothesis. Okay. Wearing different running shoes will help me run faster. Okay. Or wearing different running shoes will not help me run faster. Those are the only two possible hypotheses. Okay. Either you write the first one or the second one. Listen to me. You do not need to explain your choice in a hypothesis. Okay, you shouldn't be saying things like, it will not help me run faster because it depends on what I ate in the morning, and you know, go on and tell me your life story, right? Like you don't have to do that. Okay, you just have to say you either think it will affect it or you will not affect it. All right. Second thing, it should not be like I see some people write, the type of shoes, right? Like that's a variable, right, in your experiment, right? That's not a hypothesis. A hypothesis is a tentative answer to your question, right? Your question is, right? I guess I should say, does wearing different running shoes allow you to run faster? So what's the answer? The answer is either you think it does or it doesn't. Okay? But you can't just write it does and then or like it doesn't. Like you're not gonna get full marks, right? You have to like spell it out for me, okay? Wearing different shoes will help me run faster or it will not. Are we okay? I mean that's fine, right? Like that's why we're doing this, right? So you don't make the same mistake on the quiz. All right? All right. Let's see what else people say. Good shoes allows you to run faster. Different shoes will, yeah, I think I like the first one the most. Okay, different running shoes will make you run faster. You either say it will or it will not. That's all. Okay. All right. And then I'm gonna ask you for independent variable, right? Independent and dependent. Okay. So if you're gonna test this out, what's the independent variable? Independent variable is the type of shoes, exactly. Type of shoes, right? Like, are you wearing Nike today, Adidas tomorrow, no name brand the third day, right? Whatever, right? Okay, maybe ones with those like little, I don't know, air pockets, whatever. Okay, type of shoes. That's all I'm looking for. Dependent variable, your speed. Yeah, exactly. How fast you run, right? Speed, right? Running speed, right? So for example, you can run like 5K and then you keep track of your time, right? See how fast you run. Okay, now BB, you wrote a bunch of stuff there. Speed, distance, same track run. Only one of them is dependent variable, right? The speed, right? Not the other two, right? Not the other two. Okay. Sometimes people do this actually, not you specifically, but like on previous tests, right? Like people throw in a bunch of stuff, right? Like the expression is like you throw things onto the wall and see what sticks, right? You can't do that. Okay. If I ask you for independent variable and you write me a bunch of stuff, and in that list it happens to have type of shoes, like you're not gonna get full marks, right? Okay. You have to, just choose one, um, and hopefully it's the correct answer. Okay, what about standardized variable? What things do I have to keep the same for our experiment? Okay, good. Jay said distance you run. Good. Same person, sure, like me, right? Me or you. Same track, sure. Track is even better, right? Rather than like running on a street or something, right? Like, it's like the street could be different, right? Maybe two more. Weather, yeah. No, shouldn't be doing snowy day versus like, I don't know, rainy day. So maybe one more. Condition of person. Yeah, yeah, I know what you mean. Care to elaborate a little bit? So I know you know what you mean. I think I know what you mean, but yeah, okay. So like good, right? Time they run, right? Do I run in the morning every time, right? Okay. Same health state. Yeah. So like you want to run when you're feeling fine, right? Okay, every time. You don't want to run when you're sick versus when you're not sick, right? Maybe have the same kind of breakfast, right? Maybe have rest day in between. Okay, like you can run 5K on the first day, and then maybe if you immediately run another 5K with a different pair of shoes, like that's gonna slow you down potentially, right? Okay, you want to have rest days in between. Is that okay? So there will be a question like this on the quiz. It's not this one. I can tell you that. So like you don't have to like memorize this particular one. So it's going to be a scenario, a different scenario, and I'm going to ask you for the hypothesis. I'm going to ask you for the independent variable, dependent variable, and the standardized variable. Okay, and then the other short answer question, like I said, is going to be a picture, and then you have to describe it using qualitative quantity. Okay, try not to give me a single word. Okay, I don't like qualitative means descriptive, right? Okay, don't just say things like, you know, rough. Like, what does that mean? Like rough, okay? Even if you say it feels rough, it wouldn't probably be a right qualitative description because I'm showing you a picture, right? Like, what are you gonna do? Like, touch the screen is like, ooh, that's smooth. Like, that doesn't make sense, right? So, like, make sure the one that you're choosing, the description, is it's appropriate for the kind of observations that you are making. All right. So I think I'm going to stop there. I have done everything I could to help prepare you for the quiz. It should be okay. Okay. Historically, the class average for the first quiz is about 70 to 75, which is reasonably high, believe it or not. Yeah, just study, try your best, and I'll see you on Thursday for lecture 3. Okay. If you think of anything else you want to ask, feel free to email me. I will be sure to reply you in a timely manner. Okay, have a good afternoon.
So are we writing our